So my name is Sho Yamasaki from Osaka. So as a member of the faculty of the IFReC, so we have been working with Singapore for many years, and I really enjoy this country. And as a member, as a LIMD, so last summer, we have a joint symposium within the US. And you may see some familiar faces here. And as a director of the CiDER, so CiDER stands for the Center for Infectious Disease Education and Research. So this is not the same as the CiDER in NUS that I ran yesterday. So Center for Infectious Disease Emergencies and Response. But our mission is a convergence of knowledge for tackling infectious diseases because we learn a lot during COVID. So of course, the basic science is obviously quite important. But in the same time, as George said, the basic science itself is not sufficient for solving all problems. So for example, providing the precise information to the public and the government is really needed. And also, the professional clinicians who really knows the real infection is also needed. So to solve those problems, the CiDER was established in 2021. And we have organized 3 branches. And this year, the building, the Spaceship Earth, was established. So if you have a chance to come to Osaka, please come to CiDER as well. So today, I'd like to talk about the T cells. So of course, helper T cells are critical for the infection. And among them, the TFH is critical for B cell health to make protective antibody. However, we still don't know which kind of chronotypes and which kind of the epitope is important for the protective immunity. So the aim of our study is to identify the SARS-CoV-2-specific chronotypes expanded in recovered patients. So because to determine their epitope, because this is an important region as a T cell antigen for protection. So convalescent patient PBMC was stimulated with various antigen, and on the next day, the activated cells were sorted and applied to the single cell TCR RNA sequence to identify the TFH chronotypes. And each chronotype was reconstituted into the T cell line, the reporter cells, to co-culture with the EB transformed autologous B cells to determine the HLA and antigen epitope. So these, the TFH cluster cells was further sequenced with TCR like this. So the first, we noticed that exactly the shared chronotypes were detected across a different patient. This is unusual. So the CDR3, the TCRV usage and the CDR3 alpha is exactly identical. And the CDR3 beta chain, so there was also same, except for one amino acid. So since they are shared by convalescent patients, so we thought this might be public chronotypes. So this TCR was reconstituted in the reporter cell lines and co-culture with autologous B cells plus antigen to see the responses. And we found that the pooled S peptide vigorously activate these chronotypes. So what is the epitope? So first, between S1 and S2, S2 contains the epitope. Another commercially available selected pool was negative. So this remaining region might be possible. So some algorithm didn't work. So we tried to first determine the restricting HLA. And importantly, that this APC was mutually exchangeable, the interchangeable. So shared HLA might be likely to be responsible. So among these candidates, these 2 candidates cannot be segregated due to the linkage disequilibrium. So we separately transfected in the hex cells and finally found that the red one is the right one. So if so, that we roughly predict the hotspots. So among these 2 candidates, finally we found that this epitope is the epitope. Although these TCR have a bit different, but they can recognize the same core epitope and with a similar affinity. So we may be able to say that this is a public chronotype 017, 018. So as for the HLA, in addition to DRB1, 1501, 02, and 03, and 04 are all positive. So now we can say that DRB1, 15, whatever. So if so, this is quite frequent in East Asia and also worldwide. So we tested the public database from various ethnicities and found that the chronotypes, that this frequency was significantly higher in a COVID recovered patient. So it might have been contributed to the protective immunity. So, but this amino acid is quite different. So we suspected that any substitution might be allowed. And indeed, any substitution is allowed except for D. So we next asked whether this is structurally feasible. So Dr. Nagae had finally solved the crystal structure and found that this position 94 seems to be located away from the interface of the CDR3 alpha and the beta. So probably that's why this position is dispensable. But this is a big deal because now we can put the X, the wild card at position 94. So by this analysis, except for D, by this analysis, this p-value became extremely low, and now we can see the significant difference between mild and severe COVID patients. So this chronotype may be associated with mild symptoms. So as for the epitope, this is located here. So looking more closely, this is located with an interface of the S trimer, the forming interface. So that's why the mutation rate is quite low, and it is conserved about any kind of VOC. So next, we first filtered the chronotype by public TCR database and search for any chronotypes expanded, significantly expanded in the convalescent patients. And we found 10 chronotypes. And indeed, these are all the public chronotypes, and then they are reconstituted in the T-cell lines. And this time, to save time, we have developed the rapid epitope determination system by using semi-pooled matrix, and it worked very well. And we also determined the HLA, and indeed, this is a public. So the frequent alleles. And as for the epitope, most of the epitope was located within S2 region. So I think this is good for us. And then all epitopes are conserved across VOC. So I think this is good for us that most of the vaccine has included this epitope. So I mean the full-length spike. So the next question is, how do they acquire the publicities? There is some papers showing the cross-reactivity against the seasonal coronavirus. But in our case, no HCOVs are cross-reacted with our T cells. But instead, we found that the oral commensal bacteria possesses exactly identical core epitope +1. So indeed, this bacterial peptide can activate these T cells. And not only the peptide, the synthetic peptide, the E. coli was transformed by this antigen and co-culture with autologous antigen to present and processed naturally. And this is the bona fide cross-reactive antigen. So the cross-reactivity to symbiotic microbes, particularly with this area, may confer the resistance to SARS-CoV-2 by priming public TF8 cells in oral cavity. So this can be utilized for the booster in a post-vaccine era. So, but we have to care about the HLA diversity. So to address these questions, we have collected the HLA mismatched donors, healthy donors, with a random HLA. But all these the donors can present these peptides. So I mean, this can be, say, the promiscuous epitope. And indeed, this single peptide can activate the CD4 T cell of all donors, and also some of them are TFHs. So what about the vaccination? So of course, this vaccine is very potent, but the antibody titer was declined over time. So we collected what is called sustainers and decliners to see whether we can predict the sustainers and decliner by clonotypic tracing of the antigen-specific T cell in advance. So particularly in the case of the vaccine supplies, the limited. So in this case, we collected the proliferated cells rather than AIM. And here is the results. The conclusion that T cell responses are diverse depending on the individuals. This is quite obvious, but when we focus on the highly expanded, I mean the more than 100 cells, there is some different features. And this is quite overlapped with TFH signatures. So this signature can be established just after second shot and maintained as a memory pool. So we also determine the epitope. And then to make long story short, the early acquisition of TFH chronotypes in sustainers would be one of the characteristics of the sustainers. So even before vaccination or, of course, the infection, we do have some S-reactive T cells. So we also tested this epitope and found that here is a list of the top expanded S-reactive chronotypes before vaccination and before infection. And we found there is no TFH. And surprisingly, most of CD4 positive cells shrunk, contracted after vaccination. So like this. So, but instead, the vaccine-induced memory T cells was developed from the very rare naive T cells, probably due to the high affinity. So, and then if TFH dominant, this maybe leads to the antibody sustainers. So, early acquisition of the highly responsive TFH chronotypes may be a prerequisite for antibody longevity. So finally, we also looked at the immunodeficient patient. So I mean, XLA patient. They don't have B cells and antibodies. So this patient have infected, but the antibody titer, the virus was not eliminated for 7 months. But the symptom was not so severe like this. So that we may be able to say the T cell can control the SARS-CoV-2 to some extent. So, but around 8 months, he's running a high fever and the pneumonia, and doctor decided to use a remdesivir, and almost all the symptoms were compromised. So something have happened around this time course. The virus might be updated. So the Dr. Shioda decided to sequence the genome and found that at this time point that he found some mutation in ORF3A that it had never been identified in a public database. So this is a de novo mutation. And so we thought the T cells that recognize the original epitope might have been very effective and virus might be escaping. So indeed, this original peptide can activate PBMC to produce interferon gamma very robustly, but this mutation evade these responses. So we wanted to know that what was the evaded chronotypes. So we stimulated this PBMC with original peptide and found that this clone 111 was expanded, the higher response to this original peptide. So we cloned this TCR and reconstituted and found that the TCR actually recognized this original peptide, but it was evaded by this mutation. So we look back the single cell TCR. So what was the profile of this clone 111? We initially thought that this might be the very almighty CD8, but that is not the case. This is CD4 positive T cells. And the most characteristic the feature was a Treg signature, except for FOXP3 expression. And the CTR, the feature and the TFH feature seems to be mutually exclusive. So in the absence of humoral immunity, the resilient or flexible CD4 positive T cell might be a key to control SARS-CoV-2 infection, and escape from multifunctional CD4 positive T cells may increase the virulence. So the take-home message would be that in the absence of humoral immunity, or when the antibodies become less efficient, the virus might start to escape from efficient T cell individually. So in addition to that, we wanted to talk about the bacterial infection-specific, lipid-specific novel T cells, because Jennifer Juno kindly introduced this paper in a commentary, but due to the time limitation, I'll stop here. And thank you very much.
Sure, great talk. In the last part of your presentation, you showed that the Treg-like CD4 T cell. But in the gene signature, I found that many of the characteristic genes are the CTLA4 and the TG, and some kind of the immune exhaustion marker. So how about interpret those cells as the chronically, persistently stimulated CD4 T cell by antigen? So is it possible?
Yeah, it is possible, but in this case, we actually stimulated with peptide and so for 10 days or so, this might be the in vitro effect.
Not direct ex vivo.
Yeah.
Okay, thank you.
Thank you for the presentation. So I'm always curious about the corresponds relationship between the clones and also the T cell types, like the T cell types like the Treg. Have you tested the TFH function of your clone in vitro?
So function means to
help
the B cells produce
IL-21.
At least we tested the production of IL-21. So they produce a lot.
But actually, for this study, you mean the similar clone can appear in different types of T cells, like Treg or T.
So that's why I said this may be very flexible. So this might be good for us because too strong immune responses might be harmful. So please note that this patient are lacking B cells, antibody. So T cells have to manage all of the responses. So this kind of not too strong, not so weak, so the flexible T cell might be. But this maybe also happen in an antibody sufficient patient, particularly in human.
Yeah, especially like Treg, it is negative signal, right? But for the other activity, also, so actually the same clone can function activation, also inhibition.
Yeah, I think so.
Thank you.
Hi, nice presentation. I have a curiosity. In these patients, the one without antibody, where you said that there was this, let's say, mutation that escaped this particular response to ORF3.
Yeah, I guess so.
But these individuals have other T cells specific for other SARS-CoV-2 antigen? And if so, I mean, how can you say that everything is connected to a response to this single epitope?
So this is one of the. So we have tested several possible antigen, but this is amazing. I think because a single amino acid, a single peptide can the very robust response, interferon gamma response. So this may be a very dominant. These T cells may be dominant in this patient. So this might be quite effective to control the SARS-CoV-2 in this particular setting. And then, so that's why it is very clever for virus to target this T cell. So this is my guess.
But, sorry, these SM are other peptides?
Yeah.
Okay, thanks. So you tested also that one was a sort of dominant response.
Yeah,
okay, thank you. That was right.
Thank you. This is a really fascinating talk. I'm very interested in your the healthy PBMC with the bacteria antigen.
Oh, yeah.
I think that's a really interesting explanation. Did you look at the self-antigen? Because is there any? Because is that indication of the broader cross-reactivity? It can recognize multiple antigens, like irrelevant, including the bacteria and the self. Did you look at that? And did you look at the phenotype? I guess you did a transcriptomic analysis same time. So the phenotype of this bacteria specific T cells, are they in naive or memory phenotype? Or
yeah, that's good point. So most of those symbiotic bacterial specific antigen was Treg. So probably that's why after second vaccination they don't expand a lot. Okay. So, but as for the first case, this is an infection. So I'm not sure whether this bacterial specific the T cells were Treg or not. But I believe that this may be a TFH. So the message is a bit oxymoron, but the first part is the
really
beneficial, and the second
really
part might be
because we see the spike at the very high frequency public T cell receptor, including this one. And the second question is the cytotoxicity. So the cytotoxic molecules, and is it have any association with the CD4 response with the disease?
The last part. Yeah, we see some cytotoxic, the CTL-like signature. So recently, particularly in human, the CD4 positive CTL-like T cells are highly highlighted. So I think that this might be possible.
Okay, thank you.
Yeah.